Hello and welcome everyone to the third in the series of invited seminars by the Bangalore Developmental Biology Club. A lot of us interested in development sort of are fascinated by the origin of form and then the functions it takes on. And usually, model organisms serve as very nice, clean systems in which these kinds of questions can be answered. We have seen, especially after Suren's talk, that looking into new model systems, you are likely to see new biology. And that is the sort of spirit in which we have today's seminar, which I think is going to be very, very fascinating. And our invited speaker today is Nipam Patel, who's professor at University of Chicago and director of marine biology labs at Woods Hole. So with that, can I ask Sonia to introduce him, please?
Yes, thank you, Dhananjay, and welcome everyone who's here on Zoom, but also to those of you who are joining us from YouTube, because I know there were quite a few people who had registered, and everyone who wasn't able to attend the Zoom webinar can view it on YouTube. So welcome to all of you to, as Dhananjay said, to the third in the seminar series that the Developmental Biology Club is hosting. And we really hope that you've been enjoying the series as much as we have. So as Dhananjay said, we have Nipam Patel today. Nipam has far too many accolades and accomplishments for me to list over here, but I'll just say that he's the director of the marine biology labs at Woods Hole. A little bit about Nipam's work. He has always looked at development through the lens of evolution, and so his work has actually, looked at organisms that are traditional model organisms like Drosophila, but he's also actually developed non-traditional model organisms to ask questions about evolutionary developmental biology. And so this is, for example, means that his lab has put together genomes of the genome of a marine crustacean worm and has developed genomic tools to be able to do manipulate gene expression and observe gene expression in them. So actually, when we asked Nipam to give this talk, he asked us, what would you like me to speak about? And at that time, we, of course, told him that he should pick what he wanted to. But I guess what this means is that we can invite him again later and ask him to speak to us about the marine crustacean worm. But today, he's going to talk to us about color and how that comes to be on the butterfly wing. And of course, it happens with pigments, but what he's going to talk about is the non-pigment way in which color can come on butterfly wings. But before I actually hand it over to Nipam, there's one other thing that I want to say, because I know that this platform, the audience of the Developmental Biology Club seminar series has people from all stages of academic career. And I know that when I was reading Nipam's papers early in my development as a developmental biologist, I was equally taken by the science, but also by the aesthetics of the way he portrayed all of his science. And that made a huge impact on me. And I'm sure you're going to see that in today's seminar as well. So with that, Nipam, thank you so much for accepting our invitation to speak here. And I hope we can make this an excuse to bring you back to talk about marine crustacean worms at another time.
Thank you very much. It's a real pleasure to be able to speak to all of you. Thanks to all the organizers for the invitation. Let me go ahead and take a minute here to set up my screen share. So just 1 second here. Okay, hopefully we're good there. You see my title slide. So, as Sonia said, my lab has worked for now a number of years on quite a number of what we would say would be the non-traditional kind of organisms for the study of development, largely driven by the fact that there is a lot of really amazing biology not covered by the standard model systems. My own background is from, standard developmental model My PhD was done with primarily with Drosophila, at least the early part of it. But over the years, I've become really interested in understanding also the evolution of development. And for that, we need to look more broadly across the tree of life to understand how things like novelty has arisen, how changes in body plan have come about. And a lot of that in my lab involves a marine crustacean called Parhyella. But as Sonia said, what I decided I would talk about today is another aspect of the work that goes on in my lab, which is understanding a phenomenon called structural coloration in butterflies. So I think we'll all agree that butterflies are a fairly charismatic group of organisms. And this is a photo from part of my collection. And in all honesty, I have to admit, part of the reason I work on butterflies is I started collecting them when I was 8 years old and have amassed quite a large butterfly collection. So I always felt I also should actually use them in my science as well. And so that's one of the things that I've done in the last decade or so, is really start to explore some interesting issues in butterfly patterning. Now, there's beautiful work being done by quite a number of labs now on the genetics of how the patterns you see come to be in the wings. For example, in these heliconius butterflies right here, beautiful work by the geneticists that work on them, understanding the underlying genes that control these patterns in the wings. But what I've turned my attention to is a slightly different question, which is how colors are generated in the wing. And a very special set of colors, primarily the blues and greens. And as I'll explain, these are not generated in the standard mechanism we think of pigments, but in a phenomenon known as structural coloration. So just a little bit, we know that these patterns and colors on the wing serve quite a lot of different functions. So in addition to sort of sexual selection, which often is at work in butterflies, there's a lot of functions such as camouflage or deflecting attacks away from vital parts of the body or advertising that the butterflies are unpalatable. A lot of different reasons that, their selection acts to give these colors and patterns to butterfly wings. But where do these colors come from? So, if you zoom in on a butterfly wing, the butterfly wing is covered in these structures we call scales, and butterflies and moths belong to a group known as Lepidoptera, which means scaly wings. And so what are these scales? So these scales are a dead remnant of a cell that was growing during the pupal stage while the butterfly was in the chrysalis. And so for a cell, these are really quite large. A typical scale is about 300 microns long and maybe about 50 microns across. But it's an outgrowth of a cell that's sitting in the wing during pupal development. That cell will die, but before it dies, it secretes chitin. So this is the sugar polymer that forms the exoskeleton of all arthropods. And that chitin is very tough and lasts. And so when the butterfly emerges, these scales are these chitin-covered protrusions. So how do they get color? So if you look at the colors like these in the wings, you see that the individual scales have individual colors. That's why, if you touch a butterfly wing, you see what looks like dust on your fingers, and that dust are these scales that have come off. And an individual scale, such as you see in this example, can be these individual colors like black, brown, yellow, white. And largely the colors you're seeing here are what we call pigment colors, okay, which I'll describe in a minute. So scales come in a variety of colors and sizes and shapes and everything. But again, I'll focus on the color. So colors like red, yellow, orange, brown, black, these are the sort of what we would call the long wavelengths of the rainbow. These are based on pigments. So what do we mean by when we say pigments? So pigments are individual molecules whose structure allows them to absorb light of particular wavelengths. So here's an example just from the pigments that are in highlighters that you might use. So if you look at the yellow highlighter here, this is the organic molecule that's in that highlighter ink. And so this organic molecule, owing to this carbon structure, actually is very good at absorbing blue light. And so if you have white light shining down on it and this molecule absorbs blue, then you've got red and green being reflected, and your eye perceives that as a yellow color. So this is how pigments work. And, much of the world we think about, when we think about dyeing our clothes and things like that, it's pigments that we use. But it turns out that it's actually relatively unusual for biological molecules to be good at absorbing red and far red light. And that's what you would need to absorb if you were a pigment to be blue and green. So instead, nature's come upon a very different solution called structural color, which again, I'll describe in some detail in a minute, to create this kind of short area of the rainbow. Okay, and so these are not traditional pigments, but are created by a phenomena of light interacting with nanostructures. Now, this is not relegated to just butterflies. So this same sort of phenomena is at work, often making blues and greens in particular, in quite a number of different cases. So the iridescent blues and greens that you would see on a peacock feather, the blue that you would see on the face of a monkey, or the blue and green, the iridescent scales you might see in a fish, even human blue eyes. These are not created by pigments, but are instead created by a phenomena of light refraction. Now, I'll mention there is one big exception to this, which is why the world is so green, and that's chlorophyll. Chlorophyll is one of those rare pigments that's good at absorbing red and far red light. But back to the phenomena of structural color. So one of the best ways to illustrate structural color is really to think about something like a soap bubble. So the soap in this soap bubble doesn't have any color, yet the soap bubble in the sun will have every color of the rainbow. So how is that possible? So it's possible because of a few phenomena of light. So this is just a beam of light shining down onto a piece of glass. And this just illustrates one of those important parameters that goes on. When light hits a material of a different refractive index, so something that light is going to travel at a different speed at, like glass in this case, there's always a reflection at that surface of refractive index change. So you're seeing that some of that light is reflected, and you can calculate the amount of reflection based on the relative refractive index of the two materials. Okay, so if you have a thin material, then you have a reflection at the top surface and a reflection at the bottom surface. But remember that light also has wave-like properties. And so that means that these two waves of light now can interact with one another. Okay, so I've shown here an example. We have a very thin layer of chitin. Remember, that's that sugar polymer that arthropods make all of their exoskeleton with, and what's on the outside of the scale. And so it has a relatively high refractive index, so there's a fair bit of light reflected at both the top surface and the bottom surface of that layer of chitin. And now these waves will interact with one another. And it depends then on the distance that they've traveled, how these waves line up. So if those waves are in phase, right, after the reflection, then you will get constructive interference. The amplitude will double, and the apparent brightness of that light will actually increase fourfold. But if those waves are perfectly out of phase, for example, you'll get destructive interference, and that wavelength will disappear. Again, remember that white light is really composed of the whole rainbow of colors, and that all of these wavelengths of light, all of these colors of light have different wavelengths. So what that means is that when white light hits a surface like this, the wavelengths that undergo destructive or constructive interference vary depending on their wavelength. So you can calculate this. So for example, here, this equation's been known for a very long time. If you solve for lambda here, so the variation that's important here is the thickness of this material, and the refractive index of air, and the refractive index of chitin, and the angle that the light is hitting. So if you solve this equation, what you would get is that wavelength that undergoes maximum constructive interference, so we become much brighter. And if you take out this 1/2, you would get the wavelength that undergoes maximum destructive interference. So what that means is that certain colors will be amplified and certain colors will be suppressed. And that depends primarily then on the thickness of this layer and the and having this high refractive index difference. So butterflies can take advantage of this, and by tuning the thickness of the chitin layer, they can make any color that they want using that mechanism for those butterflies that use structural color. So what I'm going to show you next is actually a picture of a moth. This is called a sunset moth, but the only pigment color in this moth is black. And every other color you see, which is all the colors of the rainbow, are created by this phenomena of light interference. So each of their scales is actually a multi-layer of chitin and air, and each scale has a different thickness and spacing of those chitin layers. So then each individual scale can be a different color. And using that by controlling the relative thickness of those chitin layers in different areas of the wing, this butterfly or this moth can make all the colors of the rainbow using that phenomenon. Okay. So here's another very famous example. This is a blue morpho butterfly. So these butterflies have this brilliant metallic blue color to them. They're quite common in different areas of Central and South America. So this is a beautiful structural color. So there's no blue pigment in this wing. It's purely a phenomenon of light refraction. So one interesting way we can illustrate this is playing a little bit of a game with the math. So what you're going to see in this movie is we're going to drop a drop of acetone onto this wing. Now, what's going to do is replace the air in those spaces. And so normally you're going from a refractive index of 1 in air to 1.5 in chitin. But now when this drop of acetone drops on the wing, acetone has a refractive index of 1.3. So now the equations change, and the structures here, the equations are a bit more complicated. But basically what you see now is instead of blue light undergoing maximum constructive interference, it's now green light. So you see this beautiful metallic green color instead, now that the air has been replaced by acetone. So what do you see in these scales? So if you look at these scales, like every other scale, they have these ridges of chitin on them. But in the case of these blue morphos, these ridges have a remarkable substructure to them. So those are those ridges, but if we take a cross section like this across these ridges, what you see is these ridges are organized in this incredible Christmas tree pattern. And this structure is very small. So that's a 1 micron scale bar here. So the spacing between these branches of the trees and the spacing between these trees is such that there's massive constructive interference of blue light and destructive interference of most of the other wavelengths of light. So the nice thing though is, of course, we can just let that acetone evaporate back off, and now air is going to fill that space back up, and you're going to see the wing go right back to being its blue color, right? So this is a very nice way of illustrating that this is actually a structural color phenomenon. Another example I'll show you is from this little green hair streak butterfly here. So green is a very good camouflage color for butterflies and moths. But it turns out that green is very rarely a pigment in Lepidoptera, almost always created by structural phenomena. So in this case, if we zoom in on one of these green scales, so we're shining white light down on this scale, and you see what look like little facets of green light coming back at you. If you take one of these scales and break it open and look at it in SEM, what you'll see, in this case, it's not the ridges creating this color, it's these honeycomb structures, these facets that look like honeycombs down inside the scale, which are responsible for this. So this is a very specific structure called a gyroid. And I love this story. So in 1970, Alan Schoen, who's a mathematician, came up with an equation that divides space into 2 non-overlapping domains. And via this surface that's a triply periodic minimal surface. So this was a mathematical equation in the 1970s. And then it was discovered that in fact, this structure exists in the real world inside of the scale of these butterflies. And when it's made at just this size that the butterflies make it, then what you get is constructive interference of green light. And so you get, this green color and destructive interference of other wavelengths. And that's how this particular butterfly makes green. And interestingly enough, that this particular gyroid structure has evolved at least three times independently in butterflies to make green color. One more example is this emerald green swallowtail, Papilio palinuris, found throughout Southeast Asia. And so again, it's a very nice green and black color. This actually does act as sort of a camouflage. These butterflies will sit with their wings open. They'll sit in the shadows of the rainforest, and it actually helps them to blend into the background. So how do they make green? So again, if you zoom in on a scale, now you see something very different. So you see what look like dimples on the scale, and they're reflecting yellow and blue light. So how does this work? So in this case, this is a multi-layer of chitin and air, but it's curved. And that curvature is key to this phenomena. So what happens is that when white light hits the bottom of the dimple, the spacing between these layers is just right that you get constructive interference of yellow light. But light that hits the edge of the dimple is seeing a different spacing between these layers. And so you get a retroreflection of blue light. So it's this yellow plus blue that then makes you see them as being green. Now, these dimples are only a few microns across, so you can't recognize that it's yellow plus blue. You just see it as a green color. But one of the beauties of this is that the blue, because it undergoes this retroreflection, is polarized. Now they see polarized light. So presumably these butterflies do not see each other as the same color as the vegetation. So they can see each other, but they are camouflaged as green to their vertebrate predators. So these are all these examples I've given you of these structural colors in butterflies were all worked out by optical physicists. And so these optical physicists are very good at taking the wings of butterflies and then actually doing things like the TEM and the SEM analysis, doing the math, and then being able to explain to you exactly why these scales are able to produce this color. And then if they have the money as well, they often will make it out of man-made material. So this is an example with that dimple structure from the Papilio palinuris, where now it's mimicked. Instead of using chitin, it's man-made using layers of a tungsten dioxide. And so then the phenomena works the same way, and you get a green color from the light refraction, and you get actually different wavelengths from the middle versus the sides. And so that really proves that then we can understand how you can create these colors with these structures. But of course, to me as a developmental biologist, the interesting question is then how does the butterfly make a structure like this, right? So in fact, doing this out of man-made material is very difficult and not very easy for us to do it, actually relatively expensive. So how is it that a butterfly does this? And so how does it create these nanostructures that are on the order of being, at least smaller than a half wavelength of light? And how does it do it so accurately? So if it's wrong by 10 nanometers, it's the wrong color. So this is really what my lab has turned part of its attention to, is trying to understand this. So again, part of the challenge is that all of these scale structures are made in the chrysalis of the butterfly. So the imaginal disc that you have in the larvae are just the smooth epithelia without any scales, and the scale cells don't begin to differentiate until pupation. And so then what happens is those scale cells begin to grow out. And what you're seeing here is the actin cytoskeleton inside that cell. So the scale is just beginning to push out from the surface epithelia, and you see this actin cytoskeleton in it. These scales start to grow very rapidly. So now you're seeing the membranes of those scales, and you see how nicely ordered they are, like tiles across the wing. And again, you have a very ordered actin cytoskeleton inside of these scales, driving the growth of these scales. Here's an optical cross-section through a slightly older set of scales. So the pink is showing you the membrane of the scale. So you see this cross-section. And now the yellow is the filamentous actin, and you see how it's organized into these beautiful rods just underneath the surface of the membrane. And then there's additional rods on the bottom surface of the scale that are internal, that go all the way down into the cell body. So you have this very nice array of actin on the surface of the scale. So now you begin to secrete cuticle. And now in this image, you're seeing the actin again in yellow, but now you're seeing chitin being deposited in pink. And what you'll see is the chitin now starts to be deposited in those spaces between the actin rods. So those actin rods really form the cytoskeleton of the scale cell as it's growing, but then also then position those ridges by, and it may be relatively passive, that where those actin rods are underneath the membrane, you can't actually get any of the exocytosis machinery in there. So it all ends up being between those actin rods. So then actually then the next step is that the butterfly breaks down that actin and it finishes secreting chitin. So that actin cytoskeleton was there initially to grow the scale, but now with the secretion of the chitin, you have an external skeleton essentially, and then you get rid of the actin, and then you can finish secreting that chitin. So sure enough, we can show that this actin is important. We can actually depolymerize the actin using something like cytoclase D during scale development, and the scales will simply collapse. If we're in that phase when the scales have the actin cytoskeleton driving their growth, if we wait till later when the butterfly would be breaking down the actin anyway, of course, then it has an external skeleton and the scale is fine. So how does this relate to structural color? So I'll go back to this example with palinuris, that emerald green swallowtail with those dimples. So we wanted to know how this butterfly was making these dimples. How did it modify normal scale development to create this? So like every other butterfly scale, in these green scales in palinuris, you've got these nice rods of actin. Okay, and so now at a stage when it should be breaking down that actin and finishing secreting chitin, these particular green scales do something very interesting. So this is the membrane of one of those scales now at this stage. And what you're going to see, you can barely just see the dimples forming. But now you're going to see that the actin has been reorganized from rods into hexagons inside of this cell. And again, this is a single cell. And that reorganization only occurs on the rods on the top surface of the scale, not on the bottom. So again, it's a single cell. So these aren't a bunch of cells next to each other. This is a single cell that's taken those rods of actin and reorganized them as hexagons. So how might you imagine that? So, you can think of things like proteins like ARC23 that can make 70-degree angles off of rods of actin. So you might have some sort of model where you do something like that. You can imagine having proteins that might grab adjacent rods and you could get a hexagonal array. You might even imagine a situation where what you do first is just make straight connections between the actin rods and then pull on the whole array, and you might be able to get a hexagonal arrangement in that same way. So Ryan Null, who was the graduate student that did these experiments, of course, wanted to understand how you made this hexagonal pattern of actin. And so he thought, of course, we thought that an obvious way to do this would just be to dissect different stages of palinurus pupil development and find the intermediates. So Ryan set out to do that. And I have to say that, in fact, we really couldn't find convincing intermediates of this because it just seemed to be an immediate sort of phase transition. It went from rods to hexagons in sort of an instant. Now, there were some hints of some things. So you can see one of the interesting phenomena seems that you actually seem first to break down every other rod of actin, okay? And then it looked like somehow those remnants were reorganizing. Maybe that's what made the hexagons. But basically, we couldn't tell what was going on by looking at fixed samples because whatever was happening seemed to be happening very rapidly. So we decided that really what we wanted to do was to watch this phenomenon live. Now, of course, that leads to a whole slew of challenges, none, including the fact that the chrysalis is generally opaque. So how do we see into it? So we tried developing ways to grow the wings outside of the chrysalis, and Ryan actually did have some success. So here are a couple of these pupil wings now, and you're seeing them grown in a petri dish. And you can see all the colors come in as these wings are sitting here in a dish, and these are the corresponding normal adult butterfly species. The problem we discovered, though, is we could only do this successfully if we took the wings out at a certain point in pupil development. And it turned out that in all those cases that the structural colors really already were there. The pigments may come in later, but the structural components had already been built. So instead, we turned to a technique to be able to image inside of the pupae. The way we do this most commonly in the lab is we actually remove the imaginal disc for the forewing. The animal will then pupate, and it will actually not have a forewing, but it also won't have the pupil covering on that side. And so then we can see down to the normal hind wing. And this was a technique initially developed by Julian Kimura when he was actually a CA in the embryology course here at the MBL. So here you're seeing actually a movie made by Aaron Pomerantz. And this is 8 days of pupil development. So you can see the wing developing now perfectly, all the entirety of development. So now you see the scales coming in and they look kind of silvery. You see the wing veins. And now sort of the end of development, you'll see all the pigment colors come in, okay? So now we can actually watch all of wing development. And now we've been turning our attention to really being able to try to watch the actin cytoskeleton inside these living scales, inside the pupa. And so here's just a relatively preliminary movie, but you can see now the scales growing and the actin cytoskeleton forming inside of these. And this is not one of the ones that forms hexagons. So we are working to refine this technique to be able to look at it more. And it's actually a really interesting system to look at these dimples because this group of butterflies called the Acolytes swallowtails, there's 25 species or so, and they can actually make a range of colors going from a purplish blue all the way through a yellowish green. And so you might imagine a couple different ways that they could use these dimples to make different colors. So if you look here at Papilio ulysses, one of the butterflies in this group that has blue, you can see that the scales, instead of this yellow plus blue reflection, are now only having blue color. And you can see the dimples are a very different shape. They're not rounded, they have square edges. So we've been doing some things where we've been doing 3D reconstructions of the scale, and then you can see the shape of the dimples, and laying back on top of that, the optical information we have on the wavelengths of light that are being reflected in those areas. So we can begin to model the interaction of the layering of the chitin and the shape of the dimples to create the particular color. Here's an example from Papilio Paris that makes a more lime green color. And you can see that instead of dimples, it kind of has this sort of wave pattern where there's not really the cross areas of the dimples don't meet, so there's no retroreflections in there. And so you get this very different color. Now, I should also point out that there's really interesting variation that goes on at the population level as well. So there are butterflies in this group where different populations will have different colors on those areas of the wing. So there can be variation, genetic variation that creates this within a population. In addition, there's polyphenism. So the butterflies, adult butterflies, don't change the color of their scales, but in fact, there's an example here, Papilio Mackie, which has 2 generations a year, and the summer generation, the spring generation the summer generation is got this bluish color, and the spring generation has this greenish color. So this is dependent on the temperature, day-night length that they perceive as larvae. And then when they come out as adults, they can be different colors because of this. So there can be this environmentally sensitive input as well. Finally, I'll show you an example that you can also get hybridization. So this is Papilio numigeni and Papilio pericles. Now they look very different from one another, but it turns out that they're actually relatively closely related, and you can make hybrids between these butterflies. And what you get in this case is overall sort of a pattern that looks like pericles, but you can see it's a color that's intermediate. So again, this would be a fascinating, group to look at, because you could also see when you make these hybrids, how do you get these intermediate colors, or these, these combinations of colors. So that's one way that we're approaching the problem. The other way that I'll describe now is a genetic approach to looking at the phenomena of structural coloration. So for that, we use this very common butterfly in North America called the Buckeye Butterfly. So you might say, well, this doesn't seem like a very good choice to look at structural coloration, because the colors in this are orange and brown and yellow and white. All those colors we said are usually pigment colors. But it turns out that this butterfly has close relatives that live throughout the world in the same genus that in fact have very nice structural blues and greens. And so these are some examples from South America and from Asia. And so, and in fact, the Buckeye butterfly here, Hunonia guinea, is nested right inside of this group where many of these other species have beautiful structural blue and green color. So it turns out that when you collect buckeyes in North America, occasionally you do get specimens that have a little bit of blue in them. So there was a woman named Edith Smith in Florida, and she ran one of these butterfly houses where they raise butterfly chrysalises and then sell them to other butterfly places that then, will have greenhouses that people can walk through and see butterflies flying. She also would sell them for wedding releases and things like that. So she was doing it as a commercial business, and she noticed this little bit of blue, and she thought to herself, well, what if I breed together the ones with a little bit of blue? So she did this. She took the bluest individuals in each generation, and she mated them to one another for about a year, which is about 12 generations. And she was able to make the butterflies look like this instead. So this beautiful, beautiful blue color to them. And so that's Edith Fair. And Rachel Fair, then a graduate student in the lab, decided to use these butterflies to try to understand how you were making this blue structural color. So one of the things that Rachel discovered is that if you look at these blue scales, so this is a top view of the scale and a bottom view, it's really the bottom layer of the scale that's creating this blue. And in fact, if you rub the ridges off from the top, you can actually see the blue really well. And it turns out the way these butterflies create blue is that the bottom of the scale, the bottom layer of the scale, is just a flat layer of chitin, and all scales have that. But in the case of these blue scales, that layer was just the right thickness to create constructive interference of blue light. So there was a nice modeling paper that had come out already showing that if you had lamina of various thicknesses, you would in fact be able to make different colors like that. But the question was, well, how did they get to be blue? Where did they start from? So it turned out if you look at those brown scales, they actually also have a structural underpinning. So not only if you look at them from above, you see them as brown, but if you flip them over, you see this kind of gold color. And so it turns out that really they have both a pigment and a lamina that reinforces that color. So what happened? How did they change color? So it turns out that that gold color is caused by the lamina being about 100 nanometers thick, and the blue is caused by that lamina essentially getting about twice as thick. And so that switch in thickness causes a change in the colors that undergo maximum constructive interference. And that's how these butterflies, that's how Edith Smith's selection actually cause these butterflies to be blue by increasing this lamina thickness. So is that what actually goes on in normal species of these butterflies? And the answer is yes. So if you look at the scales in these other species that have nice structural blue or green, they in fact are doing it by having that lamina be the right thickness to do that. So what happens in evolution is actually was recapitulated by that selection scheme that Edith had done in the Buckeye butterflies. Now I'll show you one other interesting thing is that, so one of the predictions is that in fact, as the lamina increases in thickness, you should actually see a progressive change in the color. So at first you have a thin lamina, which actually creates this gold color because of this secondary reflection here. And then as it gets thicker, you should sort of go through a purple and then blue. And if it's really thick, you should go to green. Now I'll show you one of the things from that movie. So this is that same movie I showed you before of the live wing development in the chrysalis. But now I'm focusing in on a particular part of the wing, that butterfly, that was actually a Buckeye butterfly, and there's a region of it that has some structural bluish green. And so now if you watch this area of the movie, you see the scales, and you're gonna see it go orange and then purple and then blue and then this kind of bluish green. So even though we can't, in this particular instance, we're not directly visualizing the thickness of the lamina, we can tell you how thick the lamina is by that color shift that it's undergoing during that phase of pupil development. So then we know exactly when the lamina is getting to these different thicknesses. Now, the nice thing is that having these brown and blue butterflies that are closely related, remember now they're only separated by 12 generations of selection. Now we can use quantitative trait locus mapping to try to map the genes responsible for the blue color. So the idea is very simple. We have the blue and the brown butterflies, we mate them, we get the F1. So for every chromosome, there's an equal contribution from each of the two parents. But then we look at the F2, then we pick siblings, we make full sibling families from those F1s. So now in the F2s, of course, though, because of recombination, we have different combinations of chromosomes from the brown and the blue grandparent. So what Rachel has done is she's raised two full families, one of 501 butterflies, one of 273. I didn't even think a Buckeye could lay that many eggs, much less that anyone could raise them all to adulthood like this. But she did that. And so she has phenotyped all of these F2 butterflies. And she has now actually finished rad sequencing all of these butterflies and has started to analyze that data to do the QTL mapping. So first, let me just show you the amazing variation in the F2. So you can see all this wonderful variation in the hue of the color that you get going from, brown all the way through a bluish green color. So we can divide up the data, the phenotypes in two different ways. We can look at ones that have similar hue. So the color of blue here is very similar in all of these, but the percent of wing that is covered by this structural color varies in these different F2s. And we can use that as our phenotype and ask them, what are the QTLs that underlie that variation? And for that, we get a very striking result. There are two prominent QTLs that actually explain almost all of this genetic variation. Somewhat by chance, we actually are pretty much certain we know what one of these loci already is. It's a gene called optics. And optics was first discovered and characterized because it was mapped as a locus that controls variation in the red color pattern in heliconia butterflies. So I should mention optics is a transcription factor. And Bob Reed's lab, when they knocked it out in butterflies, I don't know that they knew exactly what they were going to get, but they got this amazing phenomena that the butterfly suddenly became blue, that those scales. And what Rachel has shown is that in fact, what happens when you knock out optics is in fact, you increase the thickness of the lamina, and that's how it's becoming blue. So almost certainly optics then is one of the genes that shows variation in the population that you can select on to get blue. What we don't know is what this other locus is, which actually has a greater percentage of the effect on the phenotype. And so we're trying to map the gene that might be underlying this QTL. But more fascinating is if we look at the F2s in a different way, if we look at them as different hues. So these wings all have the same percentage of the wing covered by this structural color. But as you see, they vary from sort of a purplish color all the way through blue to turquoise to a greenish color. And so this represents a variation in thickness of the lamina from about 140 nanometers to about 240 nanometers. So this data is more preliminary right now, and we're still refining the genetic map. And actually, we have a lot more animals, a lot more genotype data to put in. But it does seem that there are probably about 4 loci that underlie this variation. And you'll notice that these 4 are non-overlapping with the 2 that control percent coverage. So again, we don't know what these genes are, and we're still refining the QTL analysis. But it looks like then we will be able to begin to narrow down what may be the genes that really control the precise thickness of the lamina. And so I think that'll be quite fascinating to find out. And again, there's been a lot of progress now by the Lepidoptera community in having CRISPR work. So as soon as we get candidate genes, we can go in and do that. This is an unrelated gene, but just shows you how well CRISPR works for looking at the function of genes and even being able to make mosaic clones. So if a gene is essential, you can still see patches of the wing that are mutant. So hopefully from this part of the talk, I've shown you that there seems to be 2 loci, and probably certainly one is a transcription factor, the other may well be as well, that make a big switch in the thickness of the lamina. So a major flip into being a different sort of set of wavelengths of structural color. But then there seem to be other genes that may be downstream that can more precisely control the thickness of the chitin and then can give you this variation in hue. So, and I'll just end by just mentioning one other thing that we're studying in the lab, which is also transparency in butterflies. So many butterflies and moths have evolved transparency, often as a camouflage mechanism. And so if you look, there's a lot of unrelated species of butterflies that have transparency. And as you might expect with evolution, there are many, many different mechanisms that have evolved to achieve transparency. So you might say, well, you just need to get the scales out of the way. So one way to do this would be not to make scales. Well, it turns out that that's very rare. So scales also play a very important role in keeping the wings from wetting. So they provide some hydrophobicity. So generally, you don't get rid of the scales. The most common solution is to change the scales into very thin bristles, to lower the density and to change them into bristles, and now light can pass through the wing membrane. There are some moths, for example, that have evolved perfectly transparent scales, which is quite interesting to see. There's even this example of a butterfly that made the scales smaller, but they're still scale shaped, but they actually are now vertical, so they don't block the wing membrane and you can see through them. So, but you might notice that these butterflies have different efficiency of being transparent. So something like this is very nice and transparent, and this is reflected light. But this butterfly actually has a lot of glare to it. So it turns out that just getting the scales out of the way like this isn't sufficient to get really good transparency, because now you have the same problem. You have this refractive index mismatch, you're going to get some reflection. Okay, well, you might be familiar with this effect if you have glasses. So you know that glasses can be, can have glare, but you can pay extra money, you can get an anti-glare coating on the glass, and so now you don't get glare on them. And as humans, we think we're so smart to figure things out like this. Well, at least 15 million years ago, butterflies figured this out too. And so this is a beautiful example where this wing is incredibly clear, even though now you've got light bouncing, you've got transmitted light on it. And the reason is, if you look between these bristles now, the wing is covered in these nanostructures that actually prevent the glare. So butterflies have also invented these anti-glare coatings. So we're very interested, again, from a developmental biology perspective, how they can build these kinds of nanostructures. And some work recently that Aaron Pomerantz has done in the lab illustrates that these are made out of both chitin and wax, and that both of those things contribute to this anti-glare effect. So I'll end there and just thank the people that did the work. So April Dinwiddie and I originally collaborated on the sort of actin cytoskeletal work and normal scale development. At that time, April was a grad student at Yale. And then Ryan is the student who did all the work on Papilio palinuris. Rachel did all the work on the blue Buckeyes. And Kyle, Aaron, and Yuriko have been working both on the live imaging and in the transparency work. And the transparency work is also a collaboration through the Human Frontier Science Program with Marianne Elias, an ecologist in France, and Serge Berthier, a physicist in France. And so with that, I'm happy to take any questions. Thank you.
Nipam, thank you so much. That was absolutely fantastic. So I know I can see that there are some questions on Zoom. So I think Bhavya is going to be doing the moderation. So Bhavya, I'll let you take over. And for everyone, please, who's in the audience, either on YouTube or here on Zoom, please feel free to put in your questions in the Q&A, and we'll take them both from YouTube as well as from here.
Thanks, Sonia. So the first question from an anonymous attendee was basically, what does M denote in the equation?
Great, yeah. So in that equation, there was one more variable, which was M. So M is just an integer, 1, 2, 3, 4, 5. So you can imagine that basically the pattern repeats. So it's a phenomena that you might be familiar with called the Newton series. So, you can solve the equation with M being 1, but then actually the same phenomena will occur when M is 2, when everything then again is doubled. So M is just is meant to just be an integer, and usually for butterflies, 1 is the integer in that equation.
Okay, thanks. The next question is from Vijay Krishnamurthy, and he is asking, there must be spatial patterning in the thickness of chitin layers so as to get different colors at different parts of the wing in the sunset moth. How does this spatial patterning emerge?
Yeah, great question. So that's, I think, where those transcription factors come in early in development. So again, like the work in heliconius butterflies and other butterflies have indicated that there's this early patterning that's happening, both some of it during late larval stages and early pupal stages, that set up very specific domains in the wing. And then those specify the kinds and colors of scales that are going to be in that domain. So it's sort of this, signaling molecules and transcription factors early in larval and early pupal development. And then those are obviously downstream genes are then affecting specific colors. So yeah, there's an underlying pre-pattern there.
The next question is from Spatika Jairaman, and she wants to know what happens in cases where both pigments and structural coloration are at work. Does the effect of pigments overrule that of structural coloration?
Yeah, beautiful question. So yeah, the two interact in really interesting ways. So you can get, for example, purple scales where it's a combination, say, of red pigment in the scale and then a blue nanostructure on the surface. And so that will give you a purplish color. And there are many butterflies that will take advantage of having both a pigment and a structural color in the same cell, in the same scale, to create the final color. So the two work together. Interestingly enough, even something like a blue morpho, there's black pigment in the scale. And that's important because there'd be backscatter of light that doesn't interact with the cuticle. And if there's a lot of backscatter, you will get sort of a whitish effect. And in fact, morphos take advantage of that. There are morphos called white morphos, a particular type of white morpho, where if you look at it, it's pearlescent. And that's because the scales don't have any black. So there's a blue sheen, but there's no black pigment. And so you get a lot of reflected light at the same time. So yeah, definitely the pigment and the structural components can interact to give really amazing combinations of colors.
The next question is from Alok Verma. Is it possible to reconstitute a hexagonal network of actin in vitro? Similarly, is it possible to RNA-seq scale cells as they're developing to look for novel actin molecules?
Yeah,
novel actin modules.
Yeah. So first of all, for the second question, so that's one of the things we're doing. We're doing singles. We hope to be able to do single cell analysis to try to understand, what may be some of the actin modifiers. Actually, one of the things we've already done is an RNA-seq experiment where we took advantage of a transparent butterfly that just has a little window with no scale growth at all. And then we can compare that to a region with scales. And so we can do, and we actually are analyzing that data now. In terms of reconstructing the hexagonal network, I mean, that's where the beauty of like going to the cell biologists who've studied actin forever is great. And so there are some really interesting things out there now about changing the network of actin. And so those are our approaches, but I don't know of any way in vitro, at least to my knowledge right now, of creating that hexagonal network.
The next question is from Carmen There is a difference in the bridge-like structures above the chitin layer between the brown and the blue. Do these bridges alter the penetrance of the light? Does this difference contribute in any way?
Yeah, so we've looked at that. So in the examples, I think you're probably referring to the Buckeye work. And so there is some variation in other structures, but we can actually, again, rub that off and show that the vast majority of the effect is just from the lamina. So there's probably other loci that's contributing some variation in that. And it is right. It probably changes a little bit about how much light gets through, and actually also the structural integrity of the scale. But that's not playing a major role in the actual color of the scale.
The next question from Lumina Anand. What is the evolutionary advantage of transparency in the butterfly wings? Wouldn't transparency throw the butterfly's body into greater light for potential predators?
Yeah, I think so. The body represents just a small fraction of the butterfly's surface, right? So most of what you would see at a distance would be the wings. And so, in fact, there is a nice paper that just came out in Ecological Monographs that's mostly from our French collaborators, showing experimentally that, the transparency really does work. When a butterfly's wings are transparent, it's harder for the predator to see it. So yeah, I mean, the body's still there and can't be transparent, at least in the butterflies we've seen. But the transparency still is a major contributor to the butterfly being invisible to its predators.
Okay, the next question from Sukriti Kapoor is, can you tell us about the function of optics? What kind of transcription factor is it? Is it conserved? Is optics involved in coloration of structures other than scales?
Yeah, so optics was first discovered in Drosophila as a gene involved actually in eye development. So it seems to be, again, one of these transcription factors that serves many functions in development, not just in giving rise to coloration patterns in the wings. And so it's expressed in lots of different cell types at different times. So it's probably like a lot of transcription factors, that it's very cell context dependent as to what it does. It seems to be used over and over again in butterflies for many different functions, both pigmentation and structural color.
Next question is, how technically challenging it is to perform CRISPR editing in butterflies?
Yeah, it's actually, it's relatively easy. It depends a little on the species of butterflies you're working on, how well they take to being injected. But, we've been able to do it with pretty good efficiency. It depends a little on the gene too, like Winte, one of the ones that I showed a picture of, that one works surprisingly well. I think partly because it may not be essential for larval development, for embryo and larval development. So you can inject it and make animals that look, at least their wings look fully mutant. So that works very nicely. Other genes that then have sort of essential functions, and the only way you're going to get a clone is to get a little patch. So for example, in another set of experiments, we did knockouts of UBX and so of other butterfly labs. And then it only works then to get small patches, because obviously a major deletion of UBX throughout the body would kill it. And so it depends a little on the gene that you're working on. But certainly with something like the Buckeye butterflies or painted ladies that are easy to raise and relatively easy to inject, the technique's pretty straightforward.
The next question is, are the coloration in eyes of insects made along similar principles as on butterfly wings?
Yeah, in one sense, yes, in another sense, no. So all of the coloration phenomena that you would find in the wing, right, those laws of physics and so on would be the same in the eye, i.e. you can use pigments and they work the same way by absorbing particular wavelengths of light. There are a few cases where eyes will also have structural color. You've probably seen like flies that have these amazing iridescent, green eyes, and that's a structural phenomena created by nanostructures on the chitin surface. And actually, even the anti-glare properties that some insect eyes have actually use those same kind of dimple structures that we find on the wing. So it makes sense that there's the same laws of physics, and so you can use some of the same general structural ideas.
So hello, maybe I'll step in and I'll say, I think Vinita had a question. So Vinita, I've unmuted. If you can go ahead.
Thanks.
And sorry, 1 second. After that, after Vinita's question, maybe we'll move to YouTube questions, and then we can come back here if there are more over here. So Vinita, go ahead, and then you can take YouTube questions.
Yes, thanks, Sonia, and thanks, Dr. Patel. It was a really fantastic talk. I have a quick, sort of, kind of a big, wide question of: do we really know what is sort of the evolutionary context of these patterning? Because it's interesting that the hybrids, right? That really threw me off. In many ways, in the sense that you start seeing that there are very nice structures and there seems to be a design, and then you make the hybrids, and that just sort of makes the whole patterning very chaotic in the sense of how these patterns may have evolved in butterflies. So the question that I have is that, do we really know if there's any sort of phylogenetic signaling in these structures, or that it's just a series of patterns that are available, and the butterflies are going to just exploit sort of based on ecological sort of factors that may be under selection. So do we really know? And I'm sure this is something work in progress, but I'm sure you have some thoughts on that. Yeah.
It's a great question, and it's one of those things that so many areas of biology, do we really understand what the selective pressures are that generate this? So how much of it is drift, right? How much of the details don't really matter, and how much does really matter? And I think that, a lot of it, frankly, we have some areas where we have some very good data that we know that this really does make a difference to, say, mate selection, or to how well it's camouflage or something, and other things where does it really matter that much than the specificity of this pattern, other than maybe recognizing self from non-self or something like that? So I don't think we know. I mean, there are some things that, it's funny, as a butterfly collector, you really can occasionally look at something in a butterfly and realize that does really work because it fools you too. So maybe it also fools a bird. Like the blue morphos are a great example, where there's bright blue, and you're like, well, this is like really a disadvantage. I mean, probably that's mostly a sexual selection color, and the males use that and they fight with one another and the whole thing. And, maybe females select them based on that. But in a strange way, it actually does work to protect them. So one of the things they'll do is they'll flash that bright blue. And then when they're being chased, they'll suddenly sit down and the underside is just brown. And then you'll just walk right by it because you keep looking for this bright blue color. So, but those are, again, maybe sort of just so stories. So you're absolutely right. I mean, sometimes we look at these things and we don't know. I should say that those hybrids I showed don't occur in nature. They're like, those butterflies occur in completely different islands, so they aren't normally hybridized.
But in terms of I'm a plant biologist, so for me, hybrids, I mean, it seems still quite, it's a normal thing for me, because I understand geographic separation, but I mean, the hybridization just means that this is possible. And if something else is preventing it, that just makes the whole, that mechanical design, it just opens up like what's possible out there,
Yeah, I would say that like those particular butterflies, they can only be hybridized by hand. So they won't normally make it.
Oh, got it. Okay.
So they were forced to hybridize. Yeah.
Okay, perfect. Thanks.
Okay, so as promised, we'll take some questions from YouTube now. Ayanti, is there anything in YouTube? And then I promise we'll come back. There are more questions here. So.
Yeah, so there's one question from YouTube, which actually I think would tie into what Krishna Megh also asked. So Bhagyashree wants to know if the change in the wing color affects mating between brown versus blue-winged, for example. And what Krishna Megh is asking is there sexual dimorphism in any of these structural colors? And if there is, and then which direction is it in?
Yeah, so I'll tackle the sexual dimorphism first. So many butterflies are sexually dimorphic, and sometimes that has spectacular effects on the structural color. Again, maybe a great example are morpho butterflies. So for some of those species, the males are the metallic blue color and the females are orange and brown. So you can definitely have that sexual dimorphism, but then you will get some morpho females that have a coating of blue on them anyway. And so some of their scales will have that structural blue. So there's a lot of sexual dimorphism. What was I'm forgetting the first part of the question now.
Yeah, so Bhagishri's asked if the change in wing color from brown to blue in the images that you did show us, if that affects mating.
Yeah, so we didn't really do any very well controlled experiments, but Rachel did feel that it may be that the blue butterflies preferred to mate with other blue butterflies. But I would say that, you know, that was just kind of a random observation that might be true, but it would be fascinating to look at. I mean, we do know that in the case of many other species of butterflies, there is good evidence that obviously those signals play an important role in picking mates.
Right, thank you. That's all from YouTube. So if we have more time, we can do more of these questions here.
Okay, fantastic. So I'm gonna unmute Vijay. Looks like Vijay has a question, and then maybe we'll take 4 more questions from here, and then we'll let Nipam take a break. After which we can head off to spatial chat for a general interaction. So 1 question from Vijay, and then 4 more from the Zoom platform. I'll unmute you. Oh, you are unmute. Okay, go ahead, Vijay.
Hi, Nipam. A great talk, which I didn't attend. I'm very sorry, I just got in. But I wanted to ask you a question for some time. You know, many years ago, I remember you mentioning that you were in search of butterfly gynandromorphs, and you found it easier to Google them than to go to museums and look at collections. But I couldn't find that paper, but I saw another paper by Abbasi and Marcus talking about new compartments which didn't refer to this. So what's happening?
Yeah, so we haven't published our paper, but we have like a huge amount of data. And so what we find is that there really is only a single compartment in the wing, a compartment boundary in the wing. So there's just the traditional anterior and posterior compartment boundary, and that as you approach the compartment boundary, clones will flatten against the boundary, which creates kind of an appearance of a subcompartment in the anterior. But we don't find, in all our gynandomorph analysis, we don't find good support for that far posterior compartment. So we have a huge amount of data from morphos and now actually from gypsy moths. So that's something we're working on. So I'd love to, discuss that with you more at some point, but it is a paper that we're working on and now have an enormous amount of data. And now actually also have some really good molecular data that is very much like Drosophila, which is that using engrailed as a marker of compartment boundary does not work, right? So you may know, like in flies, you know, when people show a third instar disc, hedgehog is actually obeying the compartment boundary, but engrailed is actually shifted anterior to the compartment boundary. So we can go through it all, but we can show you that the compartment boundary sits between the M1 and M2 veins, and we can map it very precisely.
Great. Thank you.
Sure.
So a couple more questions from the Q&A chat here. So an anonymous attendee has asked if does the QTL for hue variation blue to green arise from variation in expression levels of the transcription factor?
So it's a great question. We don't know anything about those QTLs yet. So, and we're like, you know, right now they're not, you know, the significance is not that high, but that was based on having genotyped only a fraction of the F2s. So Rachel has finished all of the genotyping now. And so she's trying to improve that genetic map and the QTL data. You know, the problem is the loci are still going to be very big and, you know, with lots of genes under them. So we'll have other tricks to try to narrow it. So we don't really know what the molecular basis is at all of those QTLs. But yeah, one could imagine there'd be a couple of ways to do it. I don't think that the level of transcription factor, it'd be interesting. It's an interesting thought whether that could control the thickness, because yeah, that's an interesting thought. I would tend not to think that it's like an all or none switch. on the thickness, and that the hue will be something maybe about the rate of secretion, the time of secretion, enzymes that modify chitin. I think a lot of, that's the beauty of a genetic approach, is that you'll just see what you get.
Okay, thank you. Debabrata Das is asking, first of all, he thanks you for this beautiful talk, and then he's asking if it is possible to culture those larval scale cells as a tissue culture.
Yeah, so again, we haven't tried dissociating the scales and growing them. I mean, we know if we try to culture the intact wing from an early stage, we can. But it's a interesting idea. Generally, the problem is that when you try to dissociate the scales, there's such big cells with these complicated structures, whether or not you can dissociate them and keep them intact. So like what we're trying now we and other labs have tried to do for single cell seq, we're actually depending on nuclei as opposed to whole cells.
Okay. Carmen asks if what regulates the even thickness of the chitin layer? For instance, what makes sure that it's not too thin or not too thick, and particularly in the dimples?
Yeah, I know. Again, a fascinating question. I mean, you look at the beauty of these structures, and you're always like dumbfounded that it can do it so nicely and so precisely. So we don't know that, right? I mean, we know that there are a lot of genes specifically involved in chitin biosynthesis. And so, the animal, in a sense of the number of genes involved, at least has a lot of control over all of that chitin biosynthesis. So yeah, that's a fantastic question. So how does it make it a uniform thickness? And one of the things that we found is that for these structural scales, the uniform thickness is often very precise. I'll mention one interesting example where it's not. There's a butterfly called the gray pansy, which is in the same group of the Buckeyes. And it's gray. And so you might think, well, that's easy. You just have a little bit of, it's basically white with a little bit of black or something, and you get a gray butterfly. The way it makes gray is like insane. So you look at it, and every scale is a little mini rainbow. So in those scales, the chitin thickness varies from the tip of the scale to the base of the scale, so that the scale is a little rainbow. But since every scale is so small and everyone is a little rainbow, it just looks white to you. But then that's one where it actually varies the thickness of chitin in a reproducible way along the scale.
Okay, that's 4 questions. We're gonna leave it at that. I'm really sorry. There are lots of exciting questions still, and I can see Krishna Megh has his. Maybe Krishna Megh, I'll let you ask a question. But just to say it to everyone, I'm gonna put a link to Spatial Chat. We'll give everyone a biology break for 10 minutes. Please take the link and join us at Spatial Chat at I think 6:50 our time. So that's 10 minutes from now. But Krishna Megh, you can go ahead and ask your question.
Thanks for accommodating this. Nipam, again, a great talk. In our lab, we follow a lot of your lab work. Also nice to see Rachel's work progress. I chatted with her when I visited your lab several years ago. And I'll be talking to you a little bit later separately as well. What really blew my mind is that a lot of species that you spoke about do occur, some of them on our campus, others in Northeast India and so on. So I hope that a lot of people can take inspiration in studying these butterflies which are all around us. My specific question about what you mentioned is you talked about various developmental changes and then structural changes that happen in scales and then coloration. Is there any understanding of whether most of those changes are also fueled by changes in genes sequences, or are they mostly just regulatory in nature, something that happens only during development, but without actually changing sequence of the genes involved?
Yeah. So it's a great question. So it comes down to, are the changes driving these differences? Are they in the coding regions? Are they actually changing the protein, or are they in regulatory regions? So I would say at the moment, we don't have enough data to say, to give you much of an answer. Though I think it is interesting, you know, a lot of the work that's gone on that obviously you're very familiar with in other species of butterflies. You know, there's been quite a number of cases where it is regulatory changes, but of course there are always things that you can find that are protein coding. I mean, I think, you know, they're certainly in mammalian coloration, there's some beautiful examples of where it's actually in the protein coding region that's actually causing the changes. So I think as it were, the jury's still out on is one, you know, more predominant than the other. I think it also depends on the way we look for the genes. You know, we're more likely to find regulatory changes if we're looking for changes in gene expression, right? So I think we have a bias like that. So it will be interesting in the example of hue, if we ever can get to the point of, you know, understanding specifically what are the 4 actual genes underlying those 4 QTLs, are those, you know, amino acid changes in some sort of biosynthetic machinery, or again, are they regulatory changes? So I don't think we have enough data right now to say that, certainly for the questions we address. And as you know, it's an outstanding question still in evolutionary biology, much debated.
Talk to you later.
Thanks.
Okay, fantastic. Thank you everyone for showing up. Thank you, Nipam, for a fantastic talk. I've dropped the link to the spatial chat. Oh, thank you to the moderators for doing a fantastic job with the Q&A on both the platforms.